I see it as my job to try to stay on time, and I see it as your job to try and make sure that I don't stay on time. But I'll try and keep short answers if you interrupt me, which I encourage you to do. So, given also it's late in the day, I'm going to show a bunch of movies, and here are the first three. So, for those of you who are familiar with Dictio Stereo, and therefore call it Dicti, you probably know all these movies. And for those of you who are not, let me just explain them to you briefly. What is shown is a time-lapse in the upper left corner of the first 10 hours of development of roughly 1 million dictiostelia that are spread out on a 17 by 13 millimeter area of soft agar. It's an agar air interface. They're starving, and as they're starving, it's not to signal to each other, the cyclic AMP. They chemotax towards each other, and that's what you're seeing. You see these regions forming, and you can see the streaming of tens of hundreds of thousands of individuals that you see there into one cluster, one mound. The movies are looping there, which is good. What's shown on the right-hand side is the same movie as on the left-hand side, just shown at half the speed and only the period of from 2 to 7 hours, and it's frame subtracted, a little bit like what Boyle was showing. And therefore you can really see these propagating waves as the dicty is moving forward. So when a dictiostelia is moving forward, it becomes slightly stressed and therefore slightly more permeable to light. And that's what becomes clear here. Much later, meaning in the period of time from 24 to roughly 52 hours, these mounds will form slugs, and you can see them move around here. I don't hesitate to call them slugs, they truly are slugs. They can merge. If each of them bump together, they will form one slug. They might also spontaneously split up into 2 slugs. This is essentially the multicellular stage of the life cycle of the dictiostelium, which is otherwise a single-celled organism when it's happy, which I'll get back to in a second. What is shown here is a wavelet analysis of the movie shown up here. You see as a function of time during the development, the first roughly 10 hours. You see how the periodicity appears. It goes from 10 minutes and it saturates at around the developmental interesting part, which is here, at around 5 minutes. That's the periodicity with which cyclic AMP is being secreted and the dicty is reacting to it. Okay. So much for the social behavior, at least for the movie of that. This is a beautiful snapshot of the different stages that dicty goes through. After the streaming, it forms a mound. The mound will sort of rise up as a finger and then either fall out over as a slug that is sensitive to temperature and light and phototax and all kinds of taxes until it finds a nice place where it stands on its end and go through further development. And the cells that were leading the slug, the first 20% will form the stalk. And you'll see this beautiful fruiting body or flower-like structure that looks like this if you get a nice electron micrograph of it. And all the cells here in the middle, the stalk cells, the former leaders, they die. They don't make it. Whereas the 80% that are in the fruiting body form this spore-like structure, and they can go on and propagate the DNA to the next generation of themselves when better times appear. Better times is something like this. Here it took a fruiting body and just dumped it on a surface that was coated by bacteria. The fruiting body will sense that. It will basically sprout out these spores that will come out of the dormant state. They'll start eating, they'll divide. It sets up this traveling wave of a feeding front, essentially wipes clean surface and keeps all bacteria on the way. So this is shown at roughly 900 times, no, exactly 900 times real time, so 1 second gets 15 minutes real time. Okay. However, you probably want to avoid going through this whole developmental cycle if you can in any way avoid it. Simply because the numbers are not exactly against you, but not straightforward, not quite for you either. 20% of you are not going to make it. So if you can, you're going to try and stay out of that phase. And to stay out of that phase, you just have to feed. Right? You have to go out and find some food. And if you're not, essentially, you're not sitting on it, you have to find it. And that means you need some kind of search strategy. Now, there is no such thing as the best search strategy. Not in nature, not in computer science, nowhere. It will all depend on the problem you're trying to solve. So when we say the best search strategy, we mean for the given conditions. So people have shown in computer science that they came up with this no free lunch theorem, which shows that there is no such thing as the best search strategy, which tells us that the strategy we come up with must somehow match the underlying problem set, which is now the distribution of food. And since the distribution of food will also be modified over time, you would probably be better off if you can also modify your search strategy as a function of time. So here showing two examples of possible distributions of food. It can even be sort of randomly scattered. You see, these are the targets, and this is our fearsome predator, Picty, going to pick up these individual bacteria that feeds on. Or you could have clusters of bacteria that you're trying to find. Now, depending on just in these two cases, there are two different strategies that will be optimal. It's quite clear what the optimal strategy is here. You just have to walk straight. If you walk straight, you never double over your own path, and therefore you never waste any time searching an area where you know as a fact that there is no food, because you've just been there. So a straight line is the best strategy in this case. In a case where you have clusters of food, a straight line is the best strategy to find a cluster. But once you hit a cluster, you better switch your strategy to something like a more local search. Otherwise, you're going to just plow through and get out of the cluster, and that is not very smart, and that's where the food was. So the whole idea of search strategies, of course, has been studied in many other systems, maybe most famously lately the case of the albatross, where people showed that extensively, published in Nature a couple of times, that there's something like, in the first major publication, a power law in the flight times, so it's a log plot of the flight duration, and then the histogram here. Then there's a log plot, and then a couple of years later on reanalysis, and quite the log plot. Anyways, that's just a warning that you should be a little careful with data analysis. There are other possible search strategies. People have come up with sort of a two-phase kind of search strategy where you will translate first, and you switch to a different stage where you're searching locally, and you either find something or you don't, and you spontaneously switch out a state again and translocate without searching. So there are plenty of models out there trying to describe search strategies. So these are all essentially, all the data that exists so far have been for reasonably large structures, such as albatross bumblebees. Bumblebees are not as big as deer, of course, but they're still not exactly microscopic. But it simply means you don't do these kind of experiments in the lab. In addition to that, you cannot easily control the environment when you do experiments like this. You have to go out and take the data where the system is found. So we thought we would try and ask all the same questions to this well-studied system of Picty, which has the great advantage that we can control the microenvironment. We can, if not exactly pattern, then at least control the density of the local food sources. So what is shown here is a single cell, the same cell before and after, presented with foods. On the left-hand side, no food, it's quite clear. The cell is, the movie is not looping, the microscope stage is being repositioned automatically whenever the cell reaches the edge of the field of view. Let me just show it again though. Oops, that's the wrong direction. So at the same playback speed on the left and right. On the right-hand side, there's a density of roughly 0.02 bacteria per square micrometer, and you can see that the cell does a pretty good job cleaning up after itself. If it needs a bacteria, it eats a bacteria. And now what we want to find out is, what does this correspond to in terms of search strategy? So we set about trying to quantify the data we collect. So first, I'll just show you the path taken. So for the first 100 minutes of the run, when there's no food, this would be the XY positions. In the presence of food, it looks like this. It is roughly the same scales here Next thing I'll show you 170 microns, and here is what, 150, 160 microns. I don't know whether you agree with me, but to me they look not quite the same. So there clearly there's some difference. Not only does the path look different, but also first simple measures such as the mean speed differ markedly here. So it drops roughly by a factor of 2 when you present nicotine with food. So just very briefly, in the case of no food, we actually went through a bunch of time series of that was done with a grad student in the lab who was for data, and I only was involved in the data analysis of that part. We found that the instantaneous direction of motion, which is depicted here in the time series, the black line is the direction at any given time. So you see how that sort of fluctuates, possibly stochastically, and then also has a slower component to it. The blue line is just drawn by hand. We can roughly describe that as a random walk of the blue line with an additional noise that we would say is the harmonic noise, which essentially is a second-order differential equation. If we do that, we get the power spectrum. We can fit that pretty well. We can also build histograms of the instantaneous angles. We can see we have a nice little Laplacian distribution of double exponential distribution of turn angles. And we can show that the turns are also anti-correlated in time, which means that the cell can actually move reasonably straight. It does that by making a left turn and a right turn, a left turn and a right turn, and that's basically how it zigzags its way forward. Now, more details can of course be found in the publication listed there. So let me show a little bit about the foraging cell now. This is the main difference between the foraging, or rather an eating and non-eating digestion. As a function of time, you can plot the number of bacteria that you eat. We can measure that directly from the time series. And what you see is one of the limitations to getting a long time series is simply that after a period of time, the cell divides. It's eaten enough, it divides. Now you're tracking, well, not exactly the cell itself, not its daughter either. They're like sisters, right? You have two new cells, and you track one of them. After a period of time, it's eaten enough, and that will divide again. I better speed up. The time series like that, with these position plots, you can measure the mean square displacement in the case of no food and food. These are double relative plots. The full line here is a slope of 2 that would correspond to ballistic motion, and we also have a dashed line here that corresponds to diffusion. Neither of these are actually quite what is taking place. So if you look at the local slope, meaning just the logarithmic derivative of the mean square displacement, you will get something that looks like this in the case of food and no food again. You see a transition from ballistic motion to something that looks like a self-avoiding random walk, and then a crossover to lack of data, but essentially also just diffusive motion. In the presence of food, you see the same kind of crossover, but this plateau seems to be dragging on for a longer time, so it's self-avoiding for a much more extended period of time. So 3 time scales, persistence of roughly 1 to 2 minutes, self-avoidance of up to 10 minutes, and then in the case of food, further self-avoidance that can basically go on forever. I'm going to skip over this one and this one and show you just what we think is the reason why we have this self-avoidance at very long time scales. So self-avoidance at very long time scales in the presence of food seems to be simply because the cell has cleaned up all the bacteria meeting on its way, and given the choice between moving into a region where there's no food and a region where there is food, it's going to move towards the food. That's what you're seeing here. You see the digestia hitting a cluster of food, and it stays on that food until it's cleaned up. It never actually exits it. It gets close to the border, but it will not exit. Given the choice, it stays on the food. 2 minutes over time for conclusions. In the growing state, we have this at least 2 different kinds of search behavior. One, when there's no food, the cell takes reasonably long and straight runs, searching for the food, trying to find the first single bacterium. And once it hits that, if it's a cluster, it'll stay on it. If it's a single bacterium, it'll just scoop it up and continue. In case of more abundant food, it'll perform some kind of local search. It doesn't move as fast. It slows down. It cleans up after itself, and stays on the food, never going back into duties. Acknowledgement, the people, the money, and thank you.
Okay, are there any questions?
How does it know to change its behavior?
Excellent question. No real idea. It certainly doesn't have a brain, right? So one of the reasons why we are studying dicty, and one of the advantages of dicty, is that if we see any kind of complicated behavior, any kind of adaptation to the environment, we at least know that it's not something it's thinking about. It can't. Right? It must all be hardwired. So one reason why one way it can change its behavior once it encounters food is simply that it doesn't have a separate mouth and face. It eats with what it's walking around. So when it encounters food, it has to engulf it, phagocytose it. And at some point it's going to get very full. And it moves by polymerization of the cytoskeleton, and if that gets too crowded in there, that's not going to be a very efficient way of moving, so it slows down. That's our idea.
But it doesn't just move slower, it changes the shape of its path.
Right, so it moves slower. That was what I will answer first. The change in path, we think, is that it does a little bit of local chemotaxis. So it can smell a very nearby bacterium, and it will send out a pseudopod to scoop up that one. And once it sends out one pseudopod, it's sort of broken its symmetry and tends to sort of try and go in that direction. Right? So this introduces an increase in the randomness, in the short-time randomness. Right? So the persistence is much shorter. The persistence is basically set by the length scale, or the density of the food that is served to you.
Okay.
Any more questions?
It looks like that the bacteria keeps on eating. Does it stop, or?
When you say bacteria, do you mean the bacteria, or do you mean the big guy?
The big guy.
The big guy, okay. Not a bacteria. But, it becomes ill-defined, because once it has eaten enough, it divides, it becomes two. But it'll essentially only pause for the briefest period of time, which is about 10-20 minutes. Oops, I'm going to start it now. Yeah. Talk to me. And now. Right, so there's a plateau here that is less than half an hour. The cell divides, and they both take off and start moving again. But it's essentially it's never full. Once it's full, it becomes two.
Just two brief questions. So first, when you showed the trajectories, the displacement, in displacement as function of time, this is the average over many trajectories, yes? Or this is just one trajectory?
The mean square displacement?
Yes.
This is just one cell.
This is just for one cell?
Just one cell.
I see.
We have a reasonably high data density. We take a picture every 5 seconds, and we take movies for several hours. So at least down here at short time lags, we have good statistics. Here, this is just noise.
And the second question is, have you measured the proliferation? That will depend on whether you have food or not.
They certainly do not grow and divide if there's no bacteria. We are doing all these experiments on essentially nutrient-free agar. So they will run around, and they will not slow down for the first 10 hours. They seem quite happy to just run around, looking for food that we know is not there. They don't. So they're searching. After some 10 hours, they tend to slow down a little bit, but they will still be fully active, most of them after 24 hours. But they don't divide, right?
So, yeah, one last question then.
Very quick. I just wondered, if these MSDs, you have a red line and a blue line, and previously you showed us the raw data and the smooth data. Does the red line correspond to the MSD of the smooth data, or is it the smooth MSD of the raw data?
Do you understand that? Let me just tell you what it is. The blue line is the raw data where the mean square space has been calculated in non-overlapping windows. The red is for both smooth and now the mean square displacement is calculated in sliding overlapping windows.
Okay.
That the data has been smooth doesn't make any difference when you go beyond a minute. That doesn't play any role. It's only up here that you can see a difference because using a non-overlapping or overlapping windows.
Okay, one last question and the rest go to the discussion.
This should be quick. What about varying food densities? Have you looked at that, or are you going to?
I did. I didn't have enough convincing data to put one plot together that I was sort of proud of. But we varied them roughly 2 orders of magnitude, or the food content 2 orders of magnitude. Very briefly, when there's just a tiny amount of food, meaning it's going to encounter one bacterium every, say, 50 micrometers, which is 5 cell diameters, the cells seem to speed up a little bit, which doesn't make sense. They get like a little drop of sugar water. It encourages them to speed up. But otherwise, they don't seem to change much of the statistics of their behavior. And then as the density increases beyond that, they will start slowing down again and become more rapid. And at the highest possible density, they don't move very much. And they're happy where they are.
Okay, so let's thank the speaker and all the speakers of this session with bacterial suspension biofilms. Of course, we just learned that it's not just Okay. Okay, we're back. So we just learned it's not just bacterial. There's some other unicellular organisms that can function and interact in cohort or sometimes individually. So one thing I would like to emphasize for physics community here that those sort of unicellular organisms that often act in cohort and or at least live nearby and sometimes cooperate is probably the most common life form on Earth. So I've heard 95% of the biomass of life are bacteria, and they are either bacterial biofilms that live on the rocks and in the soils, or they are this bacteria, the cyanobacteria that photosynthesize in the middle of the ocean. So many of those involve multiple species. Many bacterial biofilms are associated with 75 of infection. And what's really interesting is that how different sort of bunch of scientists looked at them, and how at this point we kind of start coming back to this original sort of naturalist approach. So back in 1800s, maybe 1700s, all the way to 1960s, all people could do and were doing is do this naturalist approach. Just watch them carefully, try to understand what they observed, record, and maybe analyze with some limited learning. Then the DNA revolution came about, and molecular biology came about, and genetic approaches came by, and all the microbiological community, they stopped really caring about what's really going on. They stopped in many aspects to take movies and stuff. All they were caring about sort of molecular details. Let's see what genes are doing. Let's focus on single molecule, maybe single residue, single pathways. And on the other hand, the mathematical biology community came up with some sort of quite-sophisticated phenomenological models that sort of explain in such phenomenological details what's going on, like the Siegel equations that were mentioned at the talk. And right now, with the DNA, the system approaches came back, and then we come back with a biophysical model that again start going back to those original naturalist approaches. And now we can, with the microscopy these days, we can view the single cells and even single molecules in vivo observation. So we can do the same thing that people are doing. We can see this with much higher precision. So, and what really came out from the session, from my view, it looks like physics community and biological community, they kind of look at this problem with two different angles of this. So, what the biologists want, they really, and this quote from Sidney Brenner summarizes it pretty well. They want to connect the pre and they want to connect the phenotype and genotype, and the problem faced by pre and post-genomic genetics are pretty much the same. You want to see how the genotype translates to those mentioned behaviors. So, what do the physicists want? And the joke that we used to have in Russia is just to satisfy their own curiosity with government funding, or sometimes without. We all struggle around here. But seriously, it's kind of the new mesoscopic laws. We're looking for organizing principles. We're looking for something that's really not molecule dependent. We want TAR receptor to function the same as the other receptor. On the other hand, biologists know those receptors for a reason. So the question that biologists are asking is, what's really the difference between those receptors? What's common? So it looks like it's kind of two different approaches. Some physicists want to abstract, biologists want more details. So the question is, how do we do it together? How do we try to work together and answer those questions? And I was thinking that can sort of do it. So should physicists care about the genetics? Should we care about genes? Should we care about mutations? Should biologists care about physical mechanics, hydrodynamics, elasticity, optimal surface strategy, and etc.? So I think those are very interesting questions to talk about today. So I would like to ask speakers to come back to this stage and maybe share their opinions, and everyone is welcome to participate. So what do you guys think? Are we really asking two different questions to work together, or is there any hope of not of consolidation, but sort of bringing some questions that are very interesting to both of the communities?
You should ask a biologist.
Well, I think that at least some of it has to do with the comment you made, Oleg, which is about funding. Having served on committees and written grants, I actually think that the more that I try to work outside my comfort zone, the less the funding agencies like it. I think the answer is yeah. Of course, mathematicians should care about genetics, and biologists should care about mathematics, because it's all science, right? But the question is, if, let's say, somebody like me that's made something of a career of stumbling around, not doing anything terribly well, but doing whatever interests me, if I have a difficult time getting funded, isn't it that I come back to what I'm comfortable at and what I know will work? I don't know if that's true also with the computational guys, but I know that that's true with the biology guys.
So, if there's no comment, we're open to any questions or anyone's opinion. Anyone wants to speak out? Yeah.
Yeah, so actually the last talk was sort of triggered things in my head that are going to be shown in Ian Cousins' talk on flocculation. So when you looked at your dictyostelium, those slugs are essentially made up of individuals, and it looked like there were some individuals that were essentially leading the pack towards food particles. Is that how I think about it? You mentioned lamellipodia, but this isn't a cell that has, an outreaching arm.
All right, so let's start with the slug. The slug does consist of up to 100,000 individual cells, and there are leaders. Roughly the 20% of the cells that are in the tip of the slug are those who decide where the slug is going. Now, if you take a slug like that and cut it in two, the second half will spontaneously develop a new group of leaders, and you will again have a tip.
Right.
And those, again, that tip, those are the ones that are going to die when you go through the rest of the development profile and form your fruiting body.
Yeah, no, so following the comment on the, trying to manipulate the food sources, I mean, in Ian's talk, I don't know, are you going to talk about that tomorrow, Ian? About leaders going to different food sources?
Yeah, I will.
Yeah, so I think it'll be interesting to compare with what you all see, because it looks very similar to me.
We even talked a few weeks ago in Princeton. Now, the slug doesn't actually look for food, it looks for a place, in a sense, to die, a good place to form a fruiting body. It's only the individual cells that are known to directly look for food. And whether they communicate when they go about their business, I don't know. I don't even know that it is known or not. It is known that they tend, depending on the species again, that individual cells tend to repel each other a little bit. But whether they send out a different signal once they find a food source, I don't know.
So just a follow-up to the, I don't know if you just, so the slugs, when you said they search for places to form fruiting bodies, but if they find food instead, do the individual amoebas branch out and start duplicating again?
So I didn't talk about that now, because I don't know, essentially. I think they pretty much become dedicated to forming a fruiting body, and it takes a rather strong clue for them to change their opinion and go back to being individuals that just in the market. I certainly know that the fruiting body has a certain resistance. Once you form a fruiting body, it will wait for I think 8-12 hours before you can challenge it and make it burst open and start over again, even if presented with good stuff shortly after it's formed its fruiting body. Somebody knows more? Go ahead. Do you know more about picture than I do?
I don't know anything about picture stealing.
It's almost like your system.
I agree, we should write a paper on that.
Are the individuals within a slug genetically identical, and what happens if you introduce heterogeneity within an experiment?
So people have done that. This turned out to be an addictive discussion, it seems. Anyway, good for me.
Everyone remembers the last talk fast, so.
Yeah. So people have done experiments where they have competed two strains against each other. One that tries to avoid forming the stalk. Remember, the stalk is those are the ones who die, right? So they essentially have cheaters in the population, and then they analyze it from a mathematical game theoretical point of view. You'll find a Nature paper from 10-20 years ago that looked at that. So the different strains do have ways of distinguishing between themselves and others. Not only do they prefer to go to themselves, unless cheaters of course, but they also try to keep out the others. So there are mechanisms of keeping yourself alive.
On the swimming near walls, what's the velocity dependence to that, or is that distribution that you came, is that independent of velocity in these low Reynolds numbers?
So in fact, in this, the velocity distribution does not even appear in this graph. So in fact, so there hasn't been a lot of experiments measuring the velocity dependence. There's been, I think, people have looked at it for E. coli, and there's a strong wall dependence. But that's, as far as I know, so the velocity, if you're swimming in circles near the wall, then you go a little slower than if you're swimming in circles or straight away from the wall. Friction goes up. In general, that leads to swimming going down. There have been a couple of papers by Rosanne Bijan, I think is her name, a microbiologist from Virginia. I think that's about it.
So, but your theory says it should be independent of velocity?
Well, my theory says that, in some sense, my theory is a far field theory. Sort of the first effect of the wall. The first effect of the wall is you think of the cell as a flow disturbance with a given strength, and then you ask the right. That's the flow that you would have away from the wall. Then you ask the question, well, what is the first effect of the wall? The first effect of the wall is for a given strength that you're applying, gives you an attraction that's proportional to this flow strength. Now, of course, what I call P, my dipole, which roughly speaking scales like the drag on the cell times the size of the cell. And the velocity is changing, the drag is changing, so that means that the strength of this attraction is also going down. The important thing is that it will always go in the same direction. Whether it goes up a factor of 2, goes down by a factor of 2. Of course,
yours fit so well to the sperm data.
Yeah, so that means that, my interpretation is that the essential physics is there, and then one thing that my data, of course, our data doesn't do so well, is that my model diverges exponentially at the wall. It's a far-field model, so strictly speaking, at z equals 0, it's infinite. So, there are things that we need to worry about within basically one body length of the wall. So the last 10 microns or 5 microns, which is the length of the cell blood, the flagellum, we don't do a good job there because our far-field, quote-unquote, approximation is not anymore.
So what are the biological implications? If you're a sperm doing whatever you're doing, how big are how far are the walls from you on average? You must be getting into narrow tubes, or?
Correct. Well, in fact, you don't even need to get into narrow tubes. This shows that even if you're in big tubes, you end up near the wall.
So that seems kind of dumb. If I was a sperm, I wouldn't want to swim, and my job was to get upstream. I wouldn't want to swim in the high viscous, high drag area. Speaking.
I really saw it. So, but in some sense, what this shows is that, and perhaps that answers, or at least addresses some of those questions. There are some physical consequences of being an organism that exploits fluid forces for swimming that you just can't avoid. And that's one example. And so whether you're a pusher, first of all, there's no force, so you're a dipole, that's always true. Whether you're pushing against the fluid or you're pulling against the fluid, you always end up near the wall. For two different reasons. One is because you're attracted, the other one because you end up crashing into the wall. But this is something that you cannot avoid. And so perhaps there's a way to design something that has no inertia that can be repelled by the wall. But basically, this is an example of sort of physics.
You could have feedback, and the sperm could say, "Oh, I don't want to be near the wall."
Of course, and then you can basically change the way you stir yourself, right?
So I'm just asking, what's the answer? When you measure, if you're a spermatologist and you're measuring it in actual, whatever the channels are called.
Well, there are different channels.
I don't know.
Fallopian tube, yes. There's the uterus.
Are they all near the surface there?
Well, that's where they stay. They wait for the ovum on the wall. So in some sense, they use the fact that there's this swing. They want to be on the wall because, as you know, so it takes. I don't know how much you know about the biology of reproduction. It takes a couple of hours. From that point of view, okay. So it takes a couple of hours between insemination for mammals, between insemination to all the way to basically where the cells are going to wait. They're going to go through the cervical mucus, through the cervix. They're going to go up the uterus. They're going to go through this entrance of the fallopian tubes, which is full of mucus, and then they're going to go up the fallopian tubes, and then they're going to wait. And they can wait there for up to, I think, something like 3 days. And they stay at the wall, and they undergo a number of chemical and physical changes, but they are on the epithelium. They stay there. And then, the encounter with the egg, if it happens, the ovum, if it happens there. So they have to be at the wall because that's where they stay there. That's where they're protected. In fact, that's the only place they're protected. Otherwise, and that's why they have to hurry up because in fact, as far as I know, and I'm not a biologist, so take this with a pinch of salt. There's the female body actually all the way until when the sperm cells are stored in the fallopian tubes attacks the sperms with as a human immune response basically all the way until they're stored inside the fallopian tubes and then they're protected. So that's why they really have to hurry up.
I think that the answer to both of these questions is yes to some extent. To me, I think because I'm more on leaning towards physics, I guess the second one, the physicists, I think, try to find some universality as to what sort of minimum rules of physics that I should put in so that, for example, I could see a collective behavior and so on. And I think that's an important kind of line of research because you find a lot of interesting things that doesn't require invoking special genetics or something to explain. And I guess a biologist who is interested in understanding a more fundamental biological phenomena would have to separate things out that would be more generically can be explained. So I think the need for both of them to understand each other is definitely there. And I guess the kind of work that Ming presented to me, where you try to do everything, is the kind of thing that you could do. That's really nice because I think the truth might be somewhere in between in that the environmental or physical factors are just as important as biological factors. And you might have a number of ways that you can explain swarming or collective behavior, but unless you identify exactly what's happening at the molecular and biological level, we never sure.
Eric, very interesting about the attraction of the bacteria to the wall. I was wondering, you briefly mentioned that the bacteria gets your theory break down when the bacteria get too close to the wall. And you said it's about one body length or one length of the flagella. Is that correct?
Well, that's what I'm saying. Whether it's correct or not. Basically, anything that's far field, there's a length scale, it's the size of the cell, and so anything that's far field is valid asymptotically far away from the cell, as the cell is the point, but roughly speaking, it should remain valid almost even quantitatively up to one body length. As soon as you're within a circle of one body length, then you actually see the detailed geometry of the cell, and then you actually have to learn about everything, the two-dimensionality, and then all the high-order effects, hydrodynamic effects that Miming talked about, like high-order singularities, etc. All of this becomes important, and then you cannot do this on a piece of paper. Okay.
And for the theory you presented, basically there are two mechanisms. One is the attractive force, and the other one is the reorientation.
Correct.
And do you expect the breakdown of the theory to occur about the same distance for both of them?
Well, they're both been derived under the same sort of regime. So yes.
Yes. And so the one question I have was, you can say that there is an attractive force by just looking at the concentration profile, but you didn't present any kind of experimental evidence in terms of the reorientation.
No.
Do you aware of anybody who observed that?
That's a good question. I don't know. The problem is that after a certain time scale, which is 1 over omega, if omega is the rotation rate, you should be done. Within some fluctuations by rotational diffusion, you should be done. So you really would have to do an experiment to somehow you can clearly see where here's t equals 0, and then from t equals 0, I can follow the evolution in time of the orientational manipulation. And this was a summer experiment, and we just didn't think of the clever way, fast way to do that. This is it would be very interesting data, but I do not know of anybody who's done that.
Thank you.
So let me ask a question now. But sort of it's coming back to the points raised. Do you really think that this is a physiologically important effect? And can you give examples? Like, for bacteria. So we talk about the sperm. For bacteria, it's a physiologically important effect. And can you think about a situation in which would be evolutionarily beneficial to be attracted to the wall versus not being attracted to the wall?
So I'm not a biologist, and so I really have a hard time telling you, frankly. I mean, so I know more. I know about reproduction because I just find this interesting. So I learn. I just the biology of it, and I learn. Like what we discussed, and I know that it could be important. I've seen some paper discuss that. Of course, the distribution of nutrients near surface is different than the distribution of nutrients away from the wall. And so, in that regard, it could be important to passively being attracted to the wall, because if the nutrient distribution is better or is more concentrated or something, then passively, without even doing any chemotherapy, that's what you end up. But beyond that, I don't. I mean, personally, I don't have a strong opinion either way, just because I'm just not a biologist. So it would be hard for me. I mean, I could say yes, it's extremely important. And in fact, every biologist really should read my paper. But no.
I wanted to ask about the second part of your talk. You had the two planes, but what happens if you just have two flagella or two cilia?
Yes.
Does your analysis apply? Because it's very different geometry.
So what's different? Okay, so there are two things that are different. Cilia is completely different. Cilia cannot move past each other. So cilia are embedded into a boundary, and they're beating, and if they don't have this degree of freedom, one can swim faster or slower than the other. So for cilia, to get a phase locking, you need to have a mechanism that, if this is cilia A, this is cilia B, this is what I said earlier, you need to have a mechanism that says, well, whatever is doing the beating in cilia A needs to take into account the fact that there's another one that creates a flow field. This flow field creates a force distribution, and so you have basically you need to have a force generation, whatever does the moments distribution, etc. That is load dependent. That is depending on the fact that if I'm here and there's a guy here that's beating very fast, it gives me a load. It's very different than this guy right here. So for cilia, you need this feedback, and there have been some papers, quite a number of papers that have demonstrated that you need this feedback.
Okay, I mean, in the E. coli, if you spin one way, right, the flagella move together, and the E. coli swims, and
Correct.
if it rotates the other way, then it tumbles.
Correct.
So how come in one case the flagella seem to be in synchrony, you rotate the other way and they're not, and allowing the thing to tumble?
Well, the equivalent here would be if I have a waveform, I'm giving you a waveform, and I'm telling you it's a waveform for which the stable situation is the in phase. If I flip the direction of the wave, which would be the same thing as if I have a helix and I rotate one way, I suddenly flip the direction in which those helices are rotating, then what was stable becomes unstable, and what was unstable becomes stable. Okay, so it's purely geometry. If you have a wave that is asymmetric formed back one way, if you flip the direction in which the wave is propagating, then the asymmetry becomes completely reversed. You have to actually flip the page to see the problem, and what was stable is now becoming unstable. And so there have been a lot of work, people in physics and fluid and solid mechanics interested about this issue of geometry, of rotating helices, how do they interact, etc. And it's essentially, it's very similar. It's not at all the same analysis, but it's very similar. A lot of it is geometry, and you can say a lot just by the geometry.
Can I add on to this? So there is a fun experiment you can do to prove what you just asked about. So if you take, say, 3 coils, like hard wire, and you make 3 coils, identical coil, and if you flip them counterclockwise, they'll rotate smoothly. They synchronize. If you rotate them clockwise, they jam. So it's an asymmetry in this helix. So it's a very fun experiment. I use this to demonstrate with my 7-year-old daughter when I go to elementary school.
But they have to be floppy wires, or?
No, the hard one. So take like the hard wire. The clothes hanger would be a good material to start with. But it's quite interesting that they really move smoothly if you do counterclockwise.
It's just geometry. It's no hydrodynamics here. Just
it is. It's just symmetry.
Mm-hmm.
It's because just like I said, it's just symmetry breaking.
So the part of it which is hydrodynamics is if one is right here, one is right there, and you start rotating them, then eventually it will end up together. This part to go from that to this part, then you need hydrodynamics. Or you just need to do it by hand, but you need something to put them together.
Okay, so any more questions?
Does the wall effect we've been talking about affect the distributions for the chemotaxi stuff from Lindy's talk?
That's a good question. I don't know.
That's related.
Yes.
I only recently actually read your paper. You're talking about the biophysical paper, right? The chemotaxis, the profile with the fit with the. I don't know the answer to that.
So in our case, we always measure our cell in the middle of the device. So it's actually the wall effect is very clear that even though we didn't positive paper, and we read your paper. But it is very clear. So, but it's we haven't varied the height. It would be interesting experiment to do to look at whether chemotaxis is influenced by the wall effect. My intuitive feeling is not, because the eventually after equilibrium is reached, they should not depend on the speed with which they are swimming. So, but it's an interesting picture.
So did you do a control? Is no gradient, and what's the station distribution look like?
No chemical gradient. It's uniform.
So you don't have a wall effect then?
I'm saying X, Y plane, but in Z plane, yes, it's always there. Yeah.
Mm-hmm.
That's a very striking.
Okay.
So this is for the try to keep this distance. This is for Roy's talk. So I was actually surprised that there are so few observables that people have figured out for all of these genes. And so I guess the question is, is there a concerted strategy that one takes to find these observables, or is it just, by happenstance? How? Yeah, and what's sort of optimal for a given gene? How do you even start to, plot that trajectory?
Observable meaning observable phenotypes?
Phenotypes, yeah.
So if I stated it that way, I misstated it a bit. It's not just observable; it's quantifiable.
Right.
And so there's lots of phenotypes where people will say, "Oh, that looks a little weird." But then if at the end of the day you have fruiting bodies with spores in it, they declare it's wild type or wild type like. So that said, most of molecular biology, microbiology, for the past couple of decades has been based. The preliminary data has been usually with some sort of screen, and so you're screening for a phenotype, and so you pull up a handful of genes that have the phenotype you desire. You sequence those genes, and those are the ones you study. The hard part, at least in Myxocaudxanthus, came up when you had the whole genome, and the gene that you've been studying for 5 years was an ECF sigma factor, and you find out you have 27 more, so you knock out all of those 27, and none of them have phenotypes.
Right. I guess what I'm asking is, in biology, are there standard screenings that people sort of use, or is it sort of anything goes? How much of it is visual, phenotypes like the ones that you're sort of describing based on visual analysis versus chemical analysis versus something else?
Right. So the data, sort of the idea, the difference between a screen and a selection. So a screen can be for anything as long as it's something that can be used to differentiate the thing you want from the thing that you don't. And so no, there's no set thing that you can use, but it has to obey the criteria so that you can use that for a screen. It just has to be something where you can grow lots of an organism and then be able to figure out very quickly which one is the one you want based on something quantifiable, usually.
Just on a related note, Itai's question about phenotype, or Roy's answer about phenotype, reminded me of I think a big problem in that I come across in interacting with biologists is that their standard of phenotype is different often. It is something just visual, like, for example, with the Bacillus subtilis biofilm. Is it wrinkled or is it not wrinkled? If it's wrinkled, it's a biofilm. If it's not wrinkled, it's some lab strain that has lost the biofilm phenotype. But, that might not be the right phenotype. The right phenotype might be, is it a solid or a fluid? Does it give or does it resist strain? And so, these questions here about physicists caring about genetics or biologists caring about physics, I think in a large part we can help each other out right there in terms of, understanding materials, properties, things. understanding what differentiates a solid from a fluid, all of these things, and honing down what we call our phenotype. I mean, you had this exact same problem, right, with asking, out of all of these different behaviors in Mixo, like, what is the right one to look for? You know, and I think this is where maybe we can all help one another tremendously without having to say whether we care about genetics. You know?
Well, I mean, the way that I describe it to my students is that the problem, if the phenotype that you're looking at isn't quantifiable, then you can't compare three things. And so the example I always use is, if you have a room full of 10 people and you ask, to rank those 10 people based on who's tallest, that's easy. But if you ask to rank them based on who's nicest, that's a little harder. If you have 2 people, you can say one's nicer than the other. But when you get 3 people, it's hard to stick the one in the middle, And so that you need numbers for. And you're right. The problem is that once, like with wrinkled or not wrinkled phenotypes, phenotypes like that, unless you come up with some quantifiable metric, it's sort of like porn, when you see it. And so, two guys looking at the same thing, they both decide that it's wrinkled, and so they it's a 1 or a 0.
Yeah.
But then when you get 3 and you say which one's more wrinkled, it's tough.
Yeah.
Not if you have a quantitative measure of wrinkling.
Exactly. That's what
exactly.
There's lots of physicists out there who know what makes a wrinkle.
Yeah, and there are probably a lot of genes in the genome that modify the wrinkling. So that's where genetics comes.
Well, this is I got this exact argument. I said in a meeting a few weeks ago. We still don't know what causes wrinkles, and Roberto Coulter says, yes, we do. It's sin I and sin R, regulatory whatever. But just because, and so I had to correct myself. I said, well, the genes that cause it. Certainly, I don't doubt that. But once you know that, what's the physics of the wrinkle? Why is it wrinkly?
Okay. No points, or it's maybe a good time to conclude. So let's thank all the speakers and all the discussion participants.